No, no, open the packet. Or maybe they can open it with eyes closed, probably not. It will be tricky. Now also hold your nose closed. Okay, breathe through your mouth and give one each. You can hand it to them.
Okay. Can I open my nose?
No, no. Okay, now taste the lollipop while holding your nose and tell us what flavor you can sense. How does it taste like?
Sweet.
Just sweet,
Yeah.
no particular flavor. Now open your nose, breathe through your nose and taste. Keep your eyes closed. Do you sense any flavors?
Yeah, but I can't put my finger on. There's a flavor, but I don't know.
Okay, some kind of fruit maybe?
Yeah, fruit, but like. I It's very like an artificial strawberry flavor.
Okay, that's something. What about Pramesh?
I'm like grapes.
Okay,
I'm not exactly.
okay. Now you can distribute this to the whole gang. They get extra ones as their reward.
Wait, can I open my eyes?
Yes, now you can open your eyes and you are pretty correct. See, strawberry, grapes. So what did you learn from it?
Multiple senses to understand the taste of flavor.
Mhm. Yeah, that's pretty much it. You can go to your seat. You can take yours and go to your seat. The experiment is over. So what you saw was just that normally you think of food as related to taste, right? And you would normally imagine that if you eat this lollipop, you are going to realize what flavor it is with your taste buds. And then suddenly the sweet taste became strawberry or grape, right? So these are different senses which are going together in your perception to generate a percept of what you are tasting or what you are seeing, even when I am speaking. Okay? You actually need to see my lips to figure out what I am saying. For example, I can say, okay, that's difficult to do. I am not a ventriloquist, but you can look up this on YouTube, where you can superimpose this video with the sound, and whether somebody is saying boast or ghost, you can hear different things based on their lip movements. So the sound is the same, but it is the lip movement which is letting you understand whether somebody is touching the lips to say "b" or not touching their lips to say "g." So you always have multiple sensory inputs coming together to form a percept. Now, how does it work from the beginning? So when they put the lollipop in their mouth, what did happen? We separated out the olfaction part. So in terms of taste, can anybody here tell me what was going on?
Taste buds.
Taste buds. And what do they have?
Receptors. Taste, sound,
Sweet and different kinds of receptors.
Yes.
Different kinds of papillae which can receive different kinds of taste molecules.
Mhm. Well, papillae are actually not involved, are not specific to taste. So that is a misconception I was brought up on. And that actually comes from Aristotle's time, that you have different parts of your tongue responsible for different tastes. That is completely wrong. Every taste bud has different receptor neurons, and they can sense different taste ends at the same time. And also, you probably remember sweet, sour, salt, and bitter, and umami. These are the 5 primary tastes, but that is also not exactly true. because you can stimulate one kind of taste bud with another tastant if you give enough concentration of it. And when they go into your brain, there is a lot of cross connections happening. So it's not like clear lines going from your taste buds into your brain. Okay? So nothing is simple in neuroscience. All right, so what is happening? At the periphery, when you touch the lollipop on your tongue, it gets dissolved in your saliva, then it diffuses, it makes contacts with your receptor neurons in the taste buds, and then receptor neurons are called receptor neurons because they have receptors. Receptors have this special kind of proteins which get activated by the chemical. So in this case, sugar molecules went to the receptor neuron and there was a receptor which is specific to sugar. So the sugar molecule attaches to it temporarily and that activates the receptor neuron and that goes through your trigeminal nerve, goes into your brain, and then in the brain, the taste center processes that information. And then when olfaction comes in, that goes to olfactory bulb to the nose. And then there are these association cortices in the brain where different sensory inputs come together and you make some form of association between the two things. Okay? That's actually an open question. How do we associate different sensory inputs to be the same object? Okay? So this is called the binding problem in neuroscience. How do you bind different parts of an entity into one single percept? So the key, yes. No, it is not that kind of association. This association is between the different senses which are all coming together. That association is associative memory. Okay. So the key to understanding the brain, how it works, is neurons. Okay. So all of you are probably familiar with the structure of a neuron in some way. So this is the standard way most textbooks describe a neuron. You have a cell body or soma which contains the nucleus, and then there are these branches called dendrites, and then you have an axon. And in case of vertebrates, mammalians especially, the axon may be covered by myelin sheath, which is formed by Schwann cells, a special kind of cell. But neurons have a lot of variation. This is an example of an invertebrate neuron. In this case, this is not this kind of bipolar or multipolar neuron. Here you have just one neurite coming out of the cell body, and then it's split into two parts. One part can be axon, another part becomes dendrite. Yeah. So this is a good model to think about for vertebrate neurons, but this is probably more common form of neurons. And if you count the number of invertebrates there are, they are predominantly of this kind. So again, to repeat, neurons can come in many different shapes and sizes. Okay. So this is a cross section through retina. These drawings are by Ramani Kahal, who is thought to be the father of neuroscience, father of modern neuroscience. So this is a cross section through retina, and this top layer, actually, the whole thing is not cross section. Each layer is a section. The top layer is of amacrine cells in the retina. These you can see are spreading out. They have also one cell body at one side, and the dendrites are coming out of that and branching out. Then here you have ganglion cells. Sorry, this one. These are ganglion cells. These have, again, peculiar structure. Their dendrites are covering multiple layers, whereas amacrine cells are mostly in a single layer. And then this is a mixture of ganglion cells and amacrine cells. Okay. And then these neurons are from the dorsal column of the spinal cord. And this is from a region called inferior olive. And this is another amacrine cell from retina. Okay. And this was lizard retina, by the way. Ramani Kahal actually cut through lots of different kinds of animals and drew their neuron structures. And again, here are some more drawings by Kahal. This is a pyramidal neuron. Actually, part of the presentation is not visible. I need to have to get over that. So, where do you see full screen here? Okay, that's better. But yeah. Okay, that was the key. Okay, so this is a pyramidal cell in rabbit cortex. Cortex is the outer surface of the brain. It stands for bark in Latin. And because it looks like a tree bark, it is called cortex. And one of the major cell types in the cortex is pyramidal cell. Similarly, people who are working with hippocampus, you probably know hippocampus also has pyramidal cell. So pyramidal cells are quite ubiquitous in vertebrate brains. Then Purkinje cell is one special kind of cell. It is located in the cerebellum. So this is particularly human cerebellar Purkinje cell. And they have this very elaborate dendritic arbor because they integrate information from a lot of presynaptic neurons, a lot of other neurons. And here is a Purkinje cell example, a couple of Purkinje cells from pigeon cerebellum. Okay? They are slightly simpler than human ones, but looks pretty similar. Okay, so all of this was done by this gentleman who was a savant and pretty much a genius. So he was a gymnast, he was an artist, as well as a neuroscientist. And he received the Nobel Prize for neuroscience sometime in the early 20th century. And this was his drawing of Purkinje cell. And this is a modern microscopic image of a Purkinje cell. So all these drawings are kind of expression of his artistic ability. And these are very accurate, as you can see. And here are some more examples of different neuronal arbors. So you can see humpback whale. This is similar to a pyramidal cell or maybe a pyramidal cell. This is also similar to a pyramidal cell. This is from a dolphin cortex. This is from an African elephant's cortex. This is from a tiger's cortex. And you can see all these neurons across all these different species look very similar, irrespective of the size of the animal or the size of the brain. Then you have retinal neuron from goldfish. This looks pretty similar to amacrine cells. Then you have a motor neuron from frog brainstem. There you got a lot of branches. Here is another neuron from chicken brainstem. The red dot is indicating the location of the soma. And then you have a neuron from zebra finch. This is a bird, a small sparrow-like bird, and their brain has different structure than mammalian brains. They don't have cortex as such, but there is a region called pallium, which does a lot of information processing in their brain. And here is another cell called the giant GABAergic neuron, which is present in the locust brain. And you can see this is the whole locust brain, the red structure. And in that, the GABAergic neuron has been marked with the fluorescent green color. And this is actually a compressed version of 3D layers. So it spans over 600 microns from bottom to top. And all those slices have been merged together to make this image. And what you can see is that, like the canonical invertebrate neuron, this is a single neurite coming out, then that branches out and innervates different regions of the brain. And this video will show you through the layers of the brain. There. So this is same as this portion. And the yellow is indicating the neurites. Okay, so here we are starting from the top of the brain, and then gradually going down. I'll play this once again. So that bottom thing was the soma. This is actually a mirror image of that image. But these are two different animals. Two different brains have the exact same neuron. So this is another thing to keep in mind when you are considering different kinds of animals. So invertebrates will often have identifiable neurons. So this neuron exists one piece on each side of the brain. Okay? There is only one neuron in each animal, only two neurons in each animal, one in each hemibrain. Okay? Whereas if you go to vertebrate systems, they do not have any identifiable neurons. If you look for pyramidal cells, there are thousands and thousands of pyramidal cells. And you cannot tell from one animal to another that this is the same neuron as that one. In the case of invertebrates, you can say this for certain neurons like this, based on what they look like, their structure, which parts they innervate, and so on. This was a confocal image. Okay, here are some more locust neurons. So in the locust brain, this region is called antennal lobe, and this is a projection neuron. So when you are smelling, you have olfactory receptor neurons in your nose, and they interact with the volatile chemical that you are smelling. So the chemical goes to the receptor neuron, and the receptor neuron has a channel, receptor channel, where the chemical attaches, and that makes the channel open. And that causes some current to flow through the neuron, and the neuron gets activated. I'll say what activated means later. But then that goes to these neurons, that stimulates this kind of neurons. So in your case, you have another type of neuron. The names are different, but the neuron receives its input here, then carries it over there and spreads it out. And this is an interneuron. So this is only located here, whereas this neuron is spanning multiple brain regions. This one is located just in one single region. These are called interneurons. Okay? They usually sit between other neurons and communicate only locally. And as you can see, these are both local interneurons in the same brain region of the locust, but they look quite different. And this is another neuron. This is called Kenyon cell, and this neuron sits over here. And actually, I'll have another picture of the locust olfactory system later. So as you can see, neurons come in many different sizes and shapes, okay? Depending on the animal, and even in the same animal, there are many different kinds of neurons. Yes. They are most likely from different loci. Yeah. Okay. Here is another set of examples. So these top row of neurons are from the blowfly eye. Blowfly is the common fly which kind of starts laying eggs on carcasses. And if you read detective novels, these forensic experts, they can tell how old a dead body is based on the stage of the blowfly maggots larva, blowfly maggots. Okay? So their eyes have many different kinds of neurons, and here you can actually see the outline of the eye. The dendrites spread out all over the structure of the eye. And then here are some neurons from worms. So C. elegans is a very popular model animal. It's a platyhelminthes? No, nematode. It's a nematode, and its neurons kind of follow its body structure because the worm itself is like a long thread. The neuron also spreads along its body and sends out dendrites on the sides. Okay? This is something else. So this is probably somewhere in a ganglion. So worms have this structure of ladder-like two lines of neurons, and they are interconnected by ganglia, where many neurons are together. And this is from another worm called a clamworm. Okay. So now that you know the diversity of neuron structures, let's go back a step to make it easier to understand. So commonly, the canonical idea about neurons is that they have 4 different regions. So this is nice to understand neurons conceptually. and it will work in most cases, but not all cases. So it gets inputs, usually in the soma or dendrites, and then the cell body of the soma integrates that input. And then there is an axon which actually is conductive. So it conducts the signal towards the output region. And at the terminal of the axons are axonal terminals or synaptic boutons, as they are called, where the axon releases neurotransmitter. So this is the output of the neuron. Okay. So this is the model, ideal case. And here is one sensory neuron where it is a pseudo bipolar neuron of the retina. Or, okay, this is showing actually skin. So a lot of skin touch sensory neurons are also pseudo bipolar or pseudo unipolar. And it receives input at its dendrites. It gets stimulated at the dendrites. Then it integrates somewhere there. That turns it into a very rapid change in the voltage, which gets transmitted along the axon and then releases neurotransmitter at the axon terminals. The same thing for motor neurons. Motor neurons take signal from the brain, motor cortex probably, and then they go out, transmit the signal to the muscles. So here the muscles are getting the output. Then you have local interneurons. They connect to different groups of neurons usually, or two different neurons. So they will take input from one set of neurons and send their output to another set of neurons. Then projection neurons, as we saw, are neurons that connect different brain regions usually. So they are projecting their output to another part of the brain. And you also have neuroendocrine cells, which release their output in the bloodstream. So when you are emotional, when you are nervous, a lot of hormones are actually being activated. And how does the neural activity turn into those hormonal signals and your blood flow control and so on? That is via this kind of scenario. Your sympathetic and parasympathetic nervous system, they have connection to your blood vessels. So they can control the constriction of your blood vessel based on the neurotransmitters they release. Okay, so far this was more about anatomy. Now we are actually coming into physiology. So for doing any science, we need to do measurements and quantification, right? And how do you measure the activity of neuron, what a neuron is doing? So that is done with electrodes. The main language of neurons is electrical. Okay? They are sending signals that are electrical signals. And in normal state, every animal cell and plant cell, they maintain a voltage difference between their outside and inside. And neurons specifically maintain a negative 70 or 60 volt in most cases, but this also varies from neuron to neuron. In the same population of neurons, there will be quite a bit of variation. And then there are specific types of neurons which have some approximately average membrane voltage. Like the GGN I showed you before, that has a resting membrane voltage of minus 55 millivolts. Okay? Whereas the Kenyan cell has something like minus 65. Lots of neurons in the cortex have minus 70 millivolt. So these are not exact numbers, but rough ballpark numbers. To measure these things, what people do is they use microelectrodes. Originally, when these techniques were developed, actually by Hodgkin, Huxley, Cole, and Curtis, these scientists, they actually used silver wires and copper wires and so on. But nowadays, people use these glass microelectrodes, where you pull a glass capillary into a very narrow needle-like structure. And then you fill it with a salt solution or something like an internal solution that mimics the cytoplasmic content. And then you insert an electrode wire into that salt solution. So that makes a conductive bridge from the cytoplasm into your electrode. And then we attach that electrode with wires into an amplifier, which measures the voltage. And the amplifier has a ground. So you are basically measuring the voltage of the neurons inside with respect to the ground. And this ground will often be on the side of the brain. So the usual recording system is Imagine this is the brain. This is a neuron, and you will perfuse the brain with ACSF, artificial cerebrospinal fluid, in case of vertebrates, or Ringer solution. They are very similar actually in case of invertebrates, and people usually adjust these solutions concentrations depending on how long the brain survives, how healthy it stays. So in this solution, you have your ground wire. This is a symbol for an op-amp, this plus, minus, and output. So they can take two inputs, and there are various modes of op-amp. I won't go into that, but basically what they can do is they can give you an output, which is an amplified version of the voltage difference between the inside this and this, these two terminals. Yeah. And you can also add another microelectrode to inject current. So you can measure the current that is going in or flowing out, as well as you can push some current in or pull current out of the cell membrane. And in this way, you can actually stimulate the neuron. Okay? So normally the neuron is polarized, it is maintaining a negative voltage inside compared to the outside. Now, if you give it a negative current, then what happens? The inside voltage will go more negative. That is called hyperpolarization. Okay? In contrast, if you give a positive current, then the voltage becomes less negative, and what you get is called depolarization. And if your current amplitude is very high, then beyond the threshold, so as you increase the current amplitude, these depolarization amplitudes will increase. But if you increase it beyond the threshold, this will turn into a very sharp rise and quick fall the voltage. This is called action potential. And here you are seeing a very zoomed in version of this. If you increase the temporal scale, then it looks like a spike, like a thorn or a straight line. So that is why this is also called spike. And when neurons are active, people actually pass this recording into speakers. Okay? Nowadays they don't, but originally one way of recording from neurons was you pass the signal, you are recording the signal, but at the same time, you send part of it into a speaker. And for each of the spike, you will hear a tat sound. Okay? And a lot of this was done around Second World War. And the scientists who did this were also involved in the war effort. So this sounds like a rifle shooting. So this is also called firing of the neuron. Now this spike can encode information. So here are a couple of examples of receptor neurons spiking. Okay. A lot of receptor neurons are actually more like this than this. So in some neurons, what happens if you inject a current? So you have zero current, then you give a step current and you hold it for some time. Okay. And depending on the current amplitude, sorry, this is not the current, my bad. This is touch stimulus. Okay. You are pressing into a touch sensor. And the micrometer scale is indicating how much movement has happened, how much stretch has happened to the receptor. And as you increase the stretch, the firing rate increases. Okay. This is a very small stretch, and you see it is occasionally firing. As you increase the stretch, then the firing rate is increasing. Okay. So this is the amplitude of the stimulus, and this is the response of the neuron. So in this case, the neuron firing rate is directly encoding the amplitude of the stimulus. Okay. But more often you will see this kind of situation. Here, the neuron is responding to a change. How many millimeters per second the movement has happened. So in this case, it is only spiking at the onset and offset of the stimulus. Okay? These dots in between, they are for time marking. So in old times, people used to mark the time. So all these are usually seen on a device called oscilloscope. Okay? So it gives you a graphical representation of the voltage over time. And the oscilloscope can have multiple channels. In one channel, you can show the neuronal output. And in another channel, you can take the output from a clock. So these ticks are those clock ticks. So they are representing time axis on your graph. Now, if you reduce the slope of this movement, so you are gently increasing the pressure, you see a very fast spike, burst of spikes here. Okay. Now, as you increase the slope, you see the firing rate goes down. And actually, as you can see here the slope is 0 and there is no activity at all. So in this way, the neuron is encoding the change in stimulus rather than the stimulus itself. And a lot of your sensory neurons do exactly that. Like most of your olfactory neurons, they spike when an odor is presented and when it is taken off. Similarly, your eyes, they are actually mostly detecting the change in illumination, not individuals. And you can test this yourself. You can focus at one spot for a long time, like meditating. And at some point, you'll see that the whole world gets blurred out. That's because you have stopped your eyes from moving. And unless the eye is moving and it is registering slightly different images, it cannot really recognize anything. Yeah. That's the speed of the stimulus. So if I place a pin on your finger, or not a pin, a blunt object, then I can either hold it in contact or I can just push it in. So this is for holding contact. This is for pushing in. Mhm. These ones, these are already touch sensors. So some touch sensors do that. And then some olfactory receptor neurons also do that. But they are a very small minority. Yeah. Mhm.
Small change that happens when the stimulus.
Yeah, to some extent, yes, at the onset. And this is not all. Some neurons get inhibited. So they will be normally spiking, but when you stimulate them, they will go silent. And they are very common in olfactory system. And then there are lots of different kinds of patterns they can generate. Okay, here is another technique of recording from the brain. This one is done via extracellular recording. Okay, in this case, we are poking into the neuron and recording the intracellular voltage. But there are techniques where you can just put an electrode near the neuron and you can measure its activity. So one popular technique for this is called tetrode recording. Okay, what we do for this is we take cold, very fine wires, like 10 micron, 20 micron diameter wires, and we wind them together. Although I'm showing this as a straight line, they will be wound together like this. Only the tips will be slightly exposed and straight. Okay. Now imagine the cross section of this thing. Okay, there are 4 tips which are at different distances from the center. Now, if you put this over the brain and this neuron spikes, there is a voltage change and there is current flowing through its cell membrane, and any voltage or current changes generate electric field. So that is basic physics, right? And that electric field will have different strengths at different distances. You all know that voltage drops with distance from the source. And what is going to happen is these 4 electrodes, they will see different shapes of the spike depending on how much distance the electrical signal had to cover. So the farthest one may be like this. Okay. And here you are seeing slightly different situation. But from these 4 shapes, you can tell which neuron or which side the neuron is located in. Imagine if there was another neuron here, for that these distances will be different. Okay. For this guy, this neuron is closest. So it may have a spike like this. Okay. So different electrodes will pick up different, slightly differently shaped signals. And from these different spike shapes, you can tell that this signal is coming from one neuron and that signal is coming from another neuron. There are a lot of algorithms that have been developed for detecting them. And these are called spike sorting. This process is spike sorting. You look at the spikes, and from that, you identify individual units that they are coming from. And one thing to note that we don't call these neurons, we call these units, because you are never sure where they're coming from. There is no guarantee that 2 neurons spikes won't be exactly the same. It is quite possible that 2 neurons are like this. Okay, they are overlapping each other. Then it will be impossible to tell them apart. So this is always tentative. You are never sure. But these intracellular recordings you can be sure of. And one way to be sure of is you can mix a dye in your salt solution and you can inject the dye into the neuron at the end of your experiment. And then afterwards you can look at the brain under a microscope and you'll see the dye. And then you know, okay, I recorded from one neuron and this particular neuron. And yes.
What would be classified as noise and what would be classified from
Usually you can tell from the shapes. Yeah, so these noise won't be like this. And there are lots of standard ways of removing noise from signal. Usually they come from electronics and electrical engineering, but noise removal, and that is called filtering. So filtering is a broad topic in electrical engineering by itself. Okay. But noise will generally have very different statistics from the signal. So this is something similar to this, but here it was done on the sensillum of an insect. So insects have these little fine hairs on their body. Those are called sensilla. Okay. So if you look at the antenna of an insect, there will be very fine hairs like this. Okay. And these hairs have sensory neurons, dendrites going in them. Okay. And what this guy did was to take an electrode and poke it here, okay, and then record the signal. This is this signal. And when you zoom in, you can see that it has a mix of 2 different shapes. Okay. And because it is a single neuron, a single electrode, you can tell that only different neurons will have different shaped signal at the electrode tip. Okay. And you can see it is pretty reproducible from experiment to experiment. As experiment to experiment, by that I mean it is the same setting, but this was one stimulus, that is another stimulus. Of course, if you remove the electrode and put it back, you have no guarantee that you will get the same signal. And then from this kind of spike, we can mark the time of the spike. So trial 1, these are the time points when the neuron spiked. Trial 2, when did it spike? Trial 3, when did it spike? So this is for cell 2, this guy. This is for cell 1. And these plots are called spike rasters. And they give you an overview of the behavior of the cell with respect to the stimulus. This black mark is indicating when the stimulus was presented. So here it was presented with cyclohexanol. This is a volatile chemical, and octanol is also a volatile chemical for smell. And from these spike rasters, you can calculate the firing rate. So in each time window, you can calculate how many spikes occurred, divide that by the number of trials, and you have the average firing rate over trials. So this way you can show that, okay, a neuron can reproducibly respond to a particular stimulus. Okay, so here cell 2 very consistently fired after cyclohexanol was presented to it, whereas cell 1 did not. But for octanol, cell 1 did respond, but cell 2 did not respond. And you can probably think of how this kind of neurons can encode the identity of different odors. Right? Already from these two plots, you can tell that this odor has very different profile than that odor in terms of neural activity it evokes. But spiking is not the only way that neurons communicate. There are many neurons which are non-spiking. They are not so common in vertebrates, but they are extremely common in invertebrates. So here are examples of rod and cone cells. So the receptors, photoreceptors in your eyes, they are non-spiking neurons. So when you present a light stimulus, normally they are depolarized. Okay, but when you present light stimulus, they get hyperpolarized. And the stronger the light stimulus is, the stronger the hyperpolarization. So as you see, the number is gradually increasing from 1 to 9. The intensity of the light is increasing, and the hyperpolarization is increasing. And here is response of the GGN to different concentrations of odor. Okay, you remember GGN, that big pretty neuron? So that is a non-spiking neuron. Okay, and it responds to every odor that you give. It's not an olfactory receptor neurons, it's far deeper in the brain. It is receiving input from other neurons which are processing the olfactory information. But then as you increase the odor concentration from 10 to the power minus 5, I think mole per liter most likely, but I'm not sure. So as you increase the concentration by orders of magnitude, its depolarization amplitude actually goes up. So this is the membrane voltage. Okay? As you present the odor, the cell membrane gets depolarized, and the amount of depolarization keeps increasing as you increase the concentration. So this is an example of analog encoding. The amplitude of your signal is getting represented by the amplitude of the response. Whereas there you are seeing digital response. The spikes are all or none. They are ones or zeros, right? So in that case, the spike amplitude doesn't vary. It is pretty constant for a neuron in a given situation. But what varies is the firing rate. Now, how does this negative potential and spiking arise? So some of you have already read about it, but remember this basic idea. The cell membrane of every animal and plant cell is made of lipid, right? And these are phospholipid. They have a phosphate group and they have glyceride tails. And glyceride is basically fat, and we know fat is hydrophobic. So they run away from water. Whereas phosphoric acid, it is a charged part and it is hydrophilic. So if you put phospholipid in water, they will naturally form a structure like this. The phosphoric acid side will go towards the outer part, connect with water, and the hydrophobic part will get away from the water and come close to each other. Okay, you can make a single layer of phospholipid, and you can make a bilayer of phospholipid. And this is very stable, and this is what cell membranes are made of. And the properties of this is very constant. Okay, the lipid bilayer's thickness and its electrical properties are something which are very reproducible. And it turns out that this has a capacitance. Everybody knows what capacitance is? Yeah? No? Okay. So the capacitance of this is 1 microfarad per centimeter square. You can memorize that. That is like an universal constant. And if you look closely at the cell membrane, it is made of lipid bilayer. And in that lipid bilayer, you have some big proteins embedded. These are macromolecules. So protein molecules which can span the whole of cell membrane, and they can have a tube-like or cylindrical gap in the center. And these are channels. Some of these are channels. Not all protein molecules on the membrane are channels, but there are special kind of protein molecules which are channels, which allow some ions to pass through. Okay, so these ion channels act like resistances. And then you have cytoplasm. Usually it keeps the voltage at minus 70 millivolt. Is there any question? No? And you can build a circuit diagram of this circuit. So a capacitance is represented with, I'm sure everybody had capacitance and resistance in high school, right? So when do you get a capacitor? When you have insulator with two conductors separating some charges, right? So there is a voltage difference V between the two plates, and you have an insulator in between. The point of the insulator is that the charges cannot pass through the insulator. So they accumulate on the two sides of the insulator. Okay? But then as you pass current through, then the charges will increase because current is basically charge moving in, and up to some point it will keep increasing. After that it will stabilize. So when the voltage difference is enough to repel the charges, and then so this part is represented by the capacitor. Then this allows ions to move in. So that is a conductor. But no conductor is without resistance. Unless you are talking about superconductors, all conductors have some resistance. And they are in parallel. So this is your capacitor plate. Then this is your channel. Okay. So that is your basic neural membrane circuit. Okay. Now, normally you'll also have some channels which are non-selective and some channels which are selective for specific ions, and some channels which are variable conductance, which depend on the voltage or something else. Okay. These are represented with an arrow on top of a resistor. And that is the equivalent circuit of a neuronal membrane. So there are sodium channels which allow sodium ion to pass through, and there are potassium channels which allow potassium ions to pass through. There is something called leak channel or just membrane resistance, which is non-selective, and then you have the membrane capacitance. Okay, if you can solve this system, then you can solve how the neuron spikes. Okay? You can basically calculate the spike amplitude and time course based on these. So these are variable. Sodium and potassium conductance depends on the voltage. So these ion channels are special. And Hodgkin and Huxley basically developed a technique called voltage clamp, and Cole also developed it independently. This was mainly Hodgkin's idea. So they experimented with squid giant axon. So it is not a giant squid. Giant squid is very different, and they live very deep in the ocean. What they did was they used to go to the market, fish market, and buy loligo or squid. And then they have a special neuron which is very thick. The axon is almost 1 millimeter in diameter. So you could just look at a microscope and pull it out with forceps. And they could also insert a very fine silver wire into the axon. So that is the squid giant axon. And they had two electrodes. One was for recording and one was for current passing. And they had this whole setup to control the thing. But what they could do was basically give it a specific voltage, like a step voltage. I want the voltage to go to, say, minus 20 millivolts, okay, and then hold it there. So then what happens? The cell membrane is held at that particular voltage for a long time. So if anything is unsteady, that can settle down. But we won't go into all the details of that today. Probably in the modeling lecture, we'll do that. But basically, because of this system, these ion channels basically, you can explain the spiking, the structure of the spike in terms of sodium and potassium ion channels. This is what Hodgkin and Huxley did over 4 papers. You can look up Hodgkin and Huxley 1952 A, B, C, and D. Okay, these are like essential classic reading. What they showed was that there are channels. They did not know what the channels were, but later people found out that there are sodium channels which are normally closed. So the pore is closed. And then when you increase the voltage at smaller voltages, the pore opens up. And what that does is it allows sodium ions to come in. And in the resting state, the cell maintains a low concentration of sodium inside and high concentration of potassium inside. Okay? Because the concentration of sodium is low inside, sodium from outside will flow in. And sodium is, what charge does it have? Positive charge. So as it comes in, it will make the voltage more positive. So the cell membrane gets depolarized. But as it gets more and more positive, the potassium channel also opens up. Now, potassium is high concentration inside. So if you open it, what happens? It will flow out. Now, potassium also carries positive charge. So the voltage will go down. And at the same time, at high voltages, there is a part of the sodium channel which comes in and blocks the pore like a gate. Okay? And that is called inactivation. So then the sodium channel cannot carry any current anymore. So as potassium flows out, the membrane voltage dips down, and then later on the sodium block goes away, sodium channel block goes away, and it comes to resting membrane potential. Okay. And the sodium and potassium channels were the first ones that were predicted. Hodgkin and Huxley had no idea about what exactly does this. They only talked about permittivity of the or permeability of the cell membrane to sodium and potassium ions. Later, people figured out that these are protein molecules which have specific structures, which have specific dynamics, and how they control the flow of ions through the cell membrane. But these kind of channels are everywhere. In all cells, actually, you will have channels. And some specific ones of them are called receptors. So you can have chemoreceptors like we talked about olfaction and taste. So chemoreceptors will have molecules attached to them and open the pore of the channel, and that will allow current to pass. Then there are photoreceptors like rod and cone cells in the eye. And what they do, they get activated by light. So you have rhodopsin in these structures in the rod and cone cells. Rhodopsin absorbs light energy, a photon, and that then creates some cascade of chemical reactions, which ultimately opens some channels. And because normally the rod and cone cells are depolarized, when the channels open, they actually get hyperpolarized. So potassium flows out of them and they get hyperpolarized. Then if you think of mechanoreceptors, they will have specific mechanical structures which are attached to other cells or around the cell. So when that is indicated by this tree. So when there is a mechanical stretch or pull, that actually physically forces the pore open, and then ions can pass through. Okay. Now this action potential that we saw in one part of the neuron, it can actually propagate along the axon. Now you can think of these sodium and potassium channels are embedded all along the axon. And you saw that the sodium channel closes down, gets blocked after you get it to high voltage. So now what happens is that if you stimulate at one part of the axon, the sodium and potassium channels open, and that increases the voltage here, and then the sodium channel closes down. But that increase in voltage can activate the sodium channels nearby, which are not open yet. And in this way, this can propagate along the axon. So wherever it has just created a spike, there is a minimum time called refractory period, which is about 1 to 5 milliseconds, within which the ion channels are not able to come back to normal state. So during that time, this won't spike again. So what will happen is that the spike will occur here, and gradually it will propagate along the axon. So this is spike propagation, and animals have evolved to control the spike propagation speed using different strategies. So one is wider neurite. In many animals, you will see giant neurons, like the squid giant axon, and cockroaches also have a giant neuron. Most insects have some giant neurons connected to their eyes. And as the diameter of the axon increases, the speed of the electrical signal also increases. The speed is inversely proportional to the square root of the diameter. Sorry, the speed is proportional to the square root of the diameter. So larger diameter means larger speed. So these are commonly used for escape because the animal has to escape very quickly. They need it to be fast. The signal, like the moment you see a predator coming and the moment you have to jump off, that time interval has to be very short. So a large axon allows you to get the signal very quickly so that you can escape. Then in humans and other vertebrates, we have myelination. So what myelination does, it prevents the ions from moving out. So it is like an insulator. Whenever there is a voltage change, all the current will pass through the cytoplasm and travel to this opening, Ravius node, where there are ion channels. So as if the action potential is jumping from node to node. Okay. So this is another way of getting faster speed and less decay of signal. Also remember, as the ion channels open and close, you are losing the ion concentration. So that also takes energy, because to maintain that ion concentration difference, there are ion pumps in the cell membrane, which are using energy to pump out and in ions of specific kinds. And these are the numbers, how fast these can be. In vertebrates, the fastest ones are 100 to 120 meters per second. And the known fastest one is in a shrimp, and it goes up to 200 meters per second. Okay, now that is how signal propagates within a neuron, okay? From one end of the neuron to another end of the neuron. But how does it propagate between neurons? Because they have to communicate. If they are going to process everything not by themselves, but in collaboration with others, then they have to communicate. And that communication happens via synapses. Okay? The way people figure this out is with two electrode recordings. So you can put an electrode at a synaptic terminals, and you can put an electrode on the postsynaptic neuron. And then you can stimulate one neuron, presynaptic neuron, and see the effect on the other neuron. Okay. So here are some examples. There can be chemical synapses which are excitatory. There can be electrical synapses and chemical synapses which are inhibitory. So in case of excitatory synapse, if you give a stimulus like a spike in the presynaptic neuron, you will see a small depolarization in the postsynaptic neuron, something like this. This is usually 5 to 10 millivolts. And this is called excitatory postsynaptic potential, or EPSP. Okay. Now, if this was inhibitory instead, what happens is when you stimulate the presynaptic neuron, the postsynaptic neuron gets hyperpolarized. There is a negative blip in its voltage. And that is called IPSP, inhibitory postsynaptic potential. And finally, there are these special kind of synapses where they don't really release any chemical, but it is a direct connection between two neurons. These are called electrical synapses or gap junctions. And in this case, there is no attenuation. This is just electrical current flowing from one neuron into another. So the signal does not attenuate much. Okay, what happens in the synapses? So this is showing a presynaptic neuron. You can think of this as a presynaptic button. The terminals are called buttons when they are like a balloon. And the postsynaptic part, this these can be spines. In many neurons, it can be like a spiny structure, like a thorn, and that is called synaptic dendritic spine or synaptic spine. So in the presynaptic side, when it is depolarized, there are voltage-gated calcium ion channels which open up, and then calcium ion passes through these channels into the presynaptic terminal, and then they give rise to a cascade of chemical processes, which then make these synaptic vesicles, which are filled with chemicals called neurotransmitters, to attach to the cell membrane and release the contents. Okay? So this neurotransmitter goes through this gap called synaptic cleft, diffuse through chemically, and then on the postsynaptic side, you have receptors. Again, receptors. There can be ion channels. There can be something else. Secondary metabotropic receptors are also there. But essentially, the neurotransmitter can attach to a receptor and make it open its pore. And then again, positive ions will flow in because the postsynaptic membrane is at a negative voltage. Okay? And then you have this EPSP because of the depolarization. Mhm. Not quite. It depends. Like it can be specific to a cell type between a pair of cell types. It can be between axons. So it can be like lots of axon bundles are going together, and you'll see they are making gap junctions between them. So yes, it is faster. It is just the voltage that is propagating. Not exactly. Sorry. They are present in everything. Every animal has some gap junctions, but the purposes may be very different. Yeah. Yes.
If the in the depolarization is taken, actually tends to the electrical synapse. Is this shift significant between the action potential of the presynaptic and the postsynaptic?
What do you mean by the shift? The temporal shift.
Yeah,
That is the time for the electrical signal to propagate. It's like,
that's the time.
yeah, like even in the same neuron, you will see this thing. Like action potential propagating along the axon. So if you have an action potential here at the point here, it will take some time to come, right? This gap junction is just like that.
So, hypothetically, could this be important for locations where you might need to signal up a bunch of neurons?
Yes, it can help with synchronization. That's right. Any other questions? How are we on time? 3:10. Oh my God. Okay, now among neurotransmitters, there are many different types and people are still discovering them. So most common is acetylcholine, and this is what is present in your neuromuscular junction. So the neuron releases acetylcholine to the muscle, and that makes the muscle contract. Then you have glutamate, aspartate, glycine, GABA. These are another bunch of neurotransmitters. You have dopamine, epinephrine, norepinephrine, oxytocin, and insulin. You have nitric oxide, carbon monoxide also acting as neurotransmitter, and even ATP can act as a neurotransmitter. So it all depends on the receptors. And note that all the people will tell you that GABAergic neurons are inhibitory and glutamatergic neurons are excitatory. The neurotransmitter itself has nothing to do with the polarity of the synapse. What has is what kind of receptors does it open, and for that receptor, what is the equilibrium potential for the ion that it lets pass. What does that mean? Like we said, the inside of the neuron has more potassium, right? Let's say this is the neuron. Now, if you open specifically the potassium channel, what will happen? Potassium will flow out. And if positive charge goes out, then the voltage will go down. Similarly, the outside has more chloride than the inside. Okay. Now, if you open the chloride channel, chloride will flow in, the neuron will get inhibited. Okay. Now, if instead you reversed the concentration, you had more chlorides here, then what will happen? Chloride will flow out, and negative charge flowing out means equivalent to positive charge coming in. So it will be positive. So ultimately, the nature of the synapse, the action of the synapse depends on these ionic concentrations and the channel which ions it is letting go. For example, in vertebrates, specifically humans and mammals, babies initially have excitatory GABA. So GABA synapses are excitatory for first 2-3 days after birth, and only later on it becomes inhibitory. So depending on what the concentrations are, the same synapse can act as excitatory or inhibitory. Mhm. Oh, cool. I didn't know that. Okay, I won't spend time on this, but you already saw that non-spiking neurons can send signals in graded manner. Then you can have the effect of multiple synapses. Multiple synapses together, when they send their input to a neuron, the neuron can do some computation. So if you look at the effect of a single synapse here, if there are multiple EPSPs coming together in a short time window, they get superimposed on one another. Okay? This is a common principle called superimposition, principle of superimposition in physics. So if you have two voltages together, at one point, they just add together. So if you have like one EPSP like this, then another EPSP like this, another like this, they all add together and form this. And when that total EPSP amplitude goes above the threshold, then you can generate a whole spike. Okay, this is temporal summation. So as if over time it is summing together the inputs and generating a spike. This is an example of spatial summation. Here in one branch you have an excitatory synapse, in another you have an inhibitory synapse. And if you measure from here, the EPSP is going positive, the IPSP is going negative, and if you add them together, you will have a reduced EPSP. And you can also have multiple synapses on the same branch, and this signal, as it propagates, it will get way more attenuated, because when the current passes through, it is facing resistance, and it also leaks out of the cell membrane because of the leak channels. Whereas at synapse 2, the EPSP will be larger. And now you can imagine if synapse 2 was an inhibitory synapse, then this IPSP could completely block the effect of the synapse 1. In this way, it can do some kind of addition. And you can actually create logic circuits based on these kind of synaptic connections, and get or get not get, based on also threshold of the neuron. And to finish with, I'll tell you about a full circuit in the insect brain. So in case of locusts, they have these antennae, right? All insects have antennae, that is their nose. And the antennae have olfactory receptor neurons, which innervate the sensilla, antennal sensilla. And when a chemical attaches to a receptor on the olfactory receptor neuron, it gets to spike, and along its axon, it sends the signal to this region called antennal lobe. Okay, and these axons actually form a bundle, and that is called the olfactory nerve or antennal nerve. And here in the antennal lobe, you have projection neurons, which project the signal to another region called mushroom body, which is like similar to our cortex for higher processing in the insect brain. And within antennal lobe, there are these local neurons, which are often inhibitory, and they also get stimulated by the olfactory receptor neurons. And in turn, they inhibit the projection neuron. And then there is a circular path of projection neurons also stimulating the local neurons. Okay. Then when the projection neurons output go to mushroom body, there are these neurons called canyon cells. Okay. And these are its dendrites. And projection neuron dendrites just span this mushroom body and they connect to canyon cells through synapses. And that can stimulate the canyon cells to become activated by the odor. And then the canyon cells send their axon into these two branches. And then there is a very large neuron, this GGN guy, which gets input from all the canyon cells. But GGN itself is a non-spiking neuron. So far, PNs were spiking neurons, ORNs were spiking neurons, KC was spiking neurons. LNs can be spiking or non-spiking. GGN is non-spiking. It gets stimulated by canyon cells. And the more canyon cells get activated by an odor, the higher the depolarization of the GGN. You saw those graded depolarization, right? And in return, it inhibits all the canyon cells. So there is a feedback, negative feedback loop. And that actually keeps the canyon cell activity to a very small subpopulation. So in this way, then after the canyon cells, there are these mushroom body output neurons which pick up the signal from canyon cells, and then they go to the motor neurons and control the movement of the animal and so on. Even easier system would be to look at escape responses. Oftentimes you will see animals have a very short circuit for escape. So it will be one neuron for looming stimulus coming to your eye, then it will go to some brain center, then it is going to a motor neuron which directly makes it fly away or run away. So that kind of system is very common, but in general, it's almost impossible to track the whole path of a signal throughout the brain, especially in vertebrates, because it is so complex. There are many, many different layers, and experimentally, it is very difficult to pin down all the neurons that are involved. But this is the basic idea. You have multiple layers of neurons which process the signal in many different ways. For example, here, when the signal comes, this interaction between PNs and LNs synchronize the PNs spiking. And when they get synchronized, if you record with an electrode here, you will see wave-like phenomenon because all of them are spiking together, going up and down. So you see oscillations in your voltage outside the brain. And similarly, when they're all parallelly active, you will see that kind of local field potential oscillations here as well. And then only a subset of canyon cells actually respond to any given odor. So with that subset, you can tell which odor it is getting. Yeah, the first test we did, that experiment, same idea. And these are the common principles across different brains because these are not tied to any particular implementation. These are general computer science or algorithmic process. How you can encode information exactly, how you can be fault tolerant if there is an error in your information, how do you handle that? How do you associate your old memories with incoming information and so on. So with that, I'll finish. Thank you. Any questions? Yeah. Mhm.
Putting the information in a particular order so that it will like manage. Like you just said, it will become a wave-like structure eventually.